Good evening, everyone, and welcome to Genomics Lite. This is our Biodiversity and Evolution series, and this week we are looking at how genomics, biodiversity and evolution are studied in the lab. We're joined by two scientists today, so we've got Michelle Strickland and Petra Korlevic, and I'll let them quickly sort of introduce themselves. Michelle, do you want to unmute and introduce yourself quickly?
Sure. Hi, I'm Michelle Strickland. I work in the Tree of Life labs at the Hinxton campus in Cambridge.
Awesome. Thanks, Michelle. And Petra, do you want to quickly introduce yourself as well?
Yes. Hi, everyone. My name is Petra, and I am a postdoc in the Parasites and Microbes department, also at the Wellcome Genome campus.
Awesome, thanks Petra. And so we'll be hearing from both Michelle and Petra tonight, who obviously both study biodiversity and evolution, but quite different. So we'll get a nice contrast between two quite different sets of lab experiences. Just to start off with them, in case you've not joined one of our sessions this evening, my name's Em, I'm the Education and Learning Officer here at the Wellcome Genome campus. And we've also got Fran Gale on the call as well, and we'll be here to just help the session run smoothly for everyone. Just to start off with, we run all of these events as a way of helping everyone explore what genomics means for our lives. And me and Fran are here to moderate the space to make sure it's safe and inclusive for everyone. We welcome you to ask questions for Michelle, Petra, and all of us this evening, but we ask you to do so respectfully to everybody. In case you've not used Zoom webinar before, there's a few different features that you can use this evening. You obviously can't use your camera or your microphone to get in touch with us. So if you do need any help, use the chat box below to let us know, and we'll attempt to help you best possible. If you do have questions for our speakers this evening, it'd be useful if you can pop these in the Q&A box, because they can help us pull out questions to ask our speakers later. And we've also got closed captions available this evening as well. You can click the CC box to get closed captions, and you can also move them around the screen so that they're not in the way of anything being presented. You can also get rid of them if you don't want them. This is the second in our series of biodiversity and evolution. So last week we looked at how genomics helps us understand biodiversity and evolution. This week we're looking at sequencing genomes old and new, and how that is done in the lab. Next week, we've got Petra back again to talk about sort of the big questions in evolution and how humans are affecting the evolution of malaria, mosquitoes and medicine. And then we'll finish off the series with a careers panel with 3 different researchers from the Darwin Tree of Life project to give you an idea of the different job roles and career routes into this field of genomics. So in normal times, we'd all be based at the Wellcome Genome campus, which is just south of Cambridge. We're all pretty much still working from home at this point, obviously as we recover from the pandemic. But we always like to know where in the world you guys are. So if you would like to, find the chat box and let us know where in the world you're from, and we'll see maybe who's furthest away. We've got someone from India and someone from Colorado. My geography is not good enough to tell which is closer, but they're both pretty far away. Scotland, Chicago, Leicester, Mauritius in the Caribbean, someone from Texas. You win the closest award. I think the furthest award is actually quite tricky this week. Awesome, we've got lots of people from across the world joining us, which is always really exciting to see. On the campus then, we've got a few different research institutes, the 2 biggest being the Wellcome Sanger Institute and the EMBL EBI. Everything we research on campus obviously has something to do with genomics, that's why that's in our name. And Wellcome Sanger uses both wet lab techniques and data techniques, whereas EMBL EBI specialises just in data techniques. On campus then, we look at lots of different areas within genomics, including human genetics, or how diseases are passed down between families, and different genetic links to different diseases. We look a lot around cancer, how cancer evolves and how we might treat it. We look at cellular genetics, we look at parasites and microbes, which Petra will be touching on a little bit today. We look at the Tree of Life project, which is what Michelle is part of, and a lot around biodata, so using the data we've collected from experiments to run sort of computer science elements to our work. A couple of things I wanted to point out just before I hand over is some resources for anyone who's doing GCSEs, A-levels or equivalents around the world. So we have our Your Genome website, which is full of resources, videos, articles, that sort of thing, obviously all related to genomics. You can sign up for our education update newsletter, where we will let you know when we're running new events just like this. And if you're particularly interested in biodiversity and evolution, the Darwin Tree of Life team has a page on I'm a Scientist, where they run live chats and also have resources and activities there as well. All of these I will pop in the chat box in a minute, and also in the Eventbrite email you'll get tomorrow, so don't worry about jotting all of this down just now. And with that, that is enough of me talking. So I will hand over to Michelle. Feel free to ask questions in the chat box or Q&A as we go.
Amazing, thanks, Em. I will just bring up my slides. Is this working? This is not working. There we go. Technology error. Can only go better from here. Cool. So good afternoon. As I said, I'm Michelle Strickland. I'm part of the core laboratory at the Tree of Life. So what that means is samples come in, we do the processing, and then the DNA goes off for sequencing. So the Darwin Tree of Life, just as a kind of reminder, our aim is to sequence every plant, animal and fungi genome, so every eukaryote in the UK and Ireland. And we're aiming for the best quality genome assemblies that we can get. So this is genomes that span the entire chromosome from end to end. And one really, I think a really interesting question really is just how many genomes this encounters. So we're sort of thinking, you know, we're looking at 3,000, 20,000, 70,000 more. Really interesting how many you think we're going to be working with.
There should be a poll on your screen that you can vote on. If you're having any issues with that, just use the chat box to make a vote. Okay, so everyone so far is going for the upper end of the scale. We have no votes for the first two answers. Split between the second two, with quite a lot more now going for 70,000 or more.
Amazing. So you guys are exactly right. We're looking at over 70,000 species. That number increases pretty much every week. So I think when I first started about 18 months ago, we were looking about 66,000 species. We're well into the 70s now. Just as a side, I thought it might be quite interesting. 3,800 and 8,200 are the number of plants and fungi respectively within the UK. So those are real numbers, but they're not the total number of species that we're looking to sequence. So I thought I'd just cover very briefly just a structure of who does what within Tree of Life. We have amazing partners across the UK and even a few international working on similar projects that are sending and collecting and sending samples to us here in Hinxton. And we have a samples management team that specialise in receiving those and storing those properly for us. As I said, I work in the lab team. So here we check and dissect those samples and we prepare them and do the RNA and the high molecular weight DNA extraction for those. I'll talk a bit about that in a second. Then our samples go through sequencing. We have 4 different sequencing processes, all of which the results are then combined by our genome assembly teams, who then reassemble the full quality genomes. And then that data is then open access. It's released to the scientific world. Anybody, anywhere can access them. And we announced that with a genome note. I think there are a few already out there now. So looking at the lab, the stuff that I do, at present we have a time-tabled extraction. So we aim to extract 70 species every single week. And this could be any kind of species, plants, insects, fungi. And as I say, we're aiming for those really large high molecular weight DNA pieces. And that is what we're looking to send to sequencing. And aside from this routine stuff, we do have to do a bit of research and development. So we're testing different extraction methods. We're looking to re-extract tricky species. Some things don't work first time. We can send them through again and they might work a second time. Some things have tricky things involved. And I'll talk a bit more about that a bit later on as well. Just as a reminder of what DNA extraction is, primarily we need to break open the cells. Every cell within a specimen has that DNA in. Some specimens are trickier than others. So plants, for example, have cell walls. We need to break those open and get at the DNA inside. We then have to separate the DNA out. So we have to remove the cell walls, cell membranes, any of the cell machinery inside there, so that we're left with just the pure DNA. We wash that through, get rid of as much of any of this contaminating material as we can, and then we elute the DNA into a small volume of liquid, and that allows us to handle it and process it for the next steps. And I've just given you an overview of just what those steps look like. That's not the precise process we use, but it is a process for getting DNA out. Here is what we call a column prep. There is a filter in the middle there. In the lab, what we use something called magnetic beads. And the DNA binds to those beads, and then they can be kind of separated out and held to one side while we remove all of the liquid and contaminants.
Michelle, someone's just asked what elution means.
Sorry, elution is just a fancy term for releasing the DNA from whichever medium we bound it to. So if we bound it to a filter, it's just eluting from that filter, the same with magnetic beads. Yeah, thank you for that. So what happens to our samples? So I think this is where we start to overlap a little bit with what Petra will be talking about later. The samples that come into our labs have all been collected. They're brand new samples, so they're fresh from the environment that they're in. They're collected, and then ideally they are flash frozen. So that could be dropping them into liquid nitrogen or putting into a freezer at -70 or -80 degrees. This very quickly freezes them solid, and that helps to preserve the DNA inside. So anything like a DNases, enzymes that break up DNA, all of that is frozen, so it's inactive. And that ideally gets us the best specimen possible. So when these samples get to our labs, firstly, we need to check them. Sometimes samples come in preservatives. They could be in ethanol. They could be in a DNA storage liquid. We need to make sure that what we see is correct. If we're expecting a hornet, like in the picture, and what we see is a piece of fish, then there's been a bit of a mix-up somewhere. So we have to make sure we're processing the right thing. The same goes for body parts. If we're processing a head, we need to be able to check that that's what we've got in the tube. If we've got a bunch of legs, that's slightly different. This is less important for the DNA side of things. The DNA is the same throughout the organism. But if you're processing RNA, you have DNA that's being transcribed in different places of the body. So what is being transcribed in the head is going to be very different from what's being transcribed in the gut. So it's useful to have that bit of information, get it correct. And the same goes for the stage of life. There's going to be a very big difference between something that's in the larval stages, like a caterpillar, compared to a full-grown moth or butterfly. So again, it's all useful information, and it's something that we check in the lab as routine. Once we're happy that what we have is the right sample, some of our samples will be dissected on dry ice, and this allows us to do the first of one of our procedures. And this allows us to get the chromosome structure. So the samples for this process go through a different extraction method. And so all we do in the lab is we just take a sample and we send that to a different team. And they generate information that allows the assembly teams later down the line to reassemble a chromosome from all the gene fragments that we produce. The remaining sample is then weighed, and that determines the process that the sample goes through initially prior to DNA extraction. So larger samples we will freeze and smash, and smaller samples, because there's a risk that we lose some of the tissue as part of the smashing process, they go straight into DNA extraction, and then they are squished and ground in lysis buffer. And in the meantime, we don't go straight from sample prep into DNA extraction. So our samples, when we're not working on them, are always stored at -70 degrees. That's the best storage. That's the best way of storing them. So a little bit about this sample prep. It's fairly straightforward. You put lysis buffer, and this is a mixture of enzymes to break down proteins, detergents to break down cell membranes. All of that goes in this tube. Sample goes in this tube, and then there's a little pestle in there that's abraded. It's quite rough, and that just helps to rip the tissue apart, get the maximum surface area. Whoops, sorry, that's my. There we go. Gives us the maximum surface area so we can get the most DNA out of these samples. So it's a very destructive process. With the larger samples, we do something called a cryoprep. And in short, this is basically taking the sample, you deep freeze it in liquid nitrogen. So our samples are already frozen at -70, but in liquid nitrogen, we're going down to about -190 degrees Celsius. So these are properly deep frozen. They're very hard. And then in short, you hit them with a big hammer. And what I've got on the next slide is a short 20-second video, which just shows the process that these samples go through. So if I can get that to play. And so this is our sample going into a bag. Our samples are already frozen at this point. So what we're doing further is we're deep freezing. So all of these processes will already have been paused. But I feel that this is quite a nice video just showing that. So the sample is then dropped into the machine in front of a solid plate, and that silver bolt is what is going to go and powder the sample. And it is just a very controlled hammering situation. It's very satisfying. And then we just collect the powdered sample at the end of it. And that is then what goes into our DNA lysis. So it's very quick, it's very clean. The nice thing about it is one sample goes into a closed system. So there's no chance that we can mix anything up or get two samples in the same tube. Can we go forwards? There we go. And this is what it looks like in the lab. This is a sample after it's been destroyed. So this was the pictures of a hornet. The sample in the tube is actually a hoverfly. And what we've got there is just bits of broken up tissue sample. You can see it just scattered throughout the bag there. This is sitting on dry ice. So again, it remains cold. It remains frozen at -70. And then the sample gets tipped into this barcoded tube at the bottom. And the barcoding just allows us to track the samples through every process. That barcode is unique. And then going forwards for the larger samples, sometimes they're too big to extract the whole sample in one go. Being very flippant, you can't extract a whole oak tree. We would need a much bigger lab for that. So we take small parts. We take about 25 milligrams. So that's 25 thousandths of a gram. It's a very, very small amount. And for a majority of samples, that's plenty of tissue to get enough DNA forward for sequencing. So I think I've alluded to this quite a lot as I've been talking, but it'd be quite interesting just to see, can we process every sample in the same way, and can we get lots of high quality DNA every time?
We've had a couple of votes for yes, but most people are going for no, which I think is, yeah, sort of what you were hinting at in the last couple of slides.
Yeah, and does anybody have any thoughts why this might be the case? I think we've got room for that going in the chat box somewhere.
Yeah, so if is there anyone that voted no, which is most people, yeah, why do we think that maybe we can't use the same method for everything?
And the more specific, the better, to be fair.
I mean, someone's asked a question that actually is sort of an answer to this. So someone's asked, are there any species that you've come across that can withstand the temperatures of -70 or yeah, don't flash freeze in the same way as other species at that temperature? So I guess that's an answer in itself, that if there were any species that could withstand that, then that wouldn't quite work the same.
So to the best of my knowledge, nothing we've come across yet can survive -70. I say this very loosely. I think if you start looking at things like tardigrades and nematodes, I think some of those you probably could wake back up afterwards. But the majority of things that we get, definitely not. Things generally do expire at -70. You can't wake them up from that. With some of the preservatives, it's been quite interesting. Samples kept in ethanol, so that's 100% pure alcohol. Sometimes they can be really tricky. When it comes to doing the cryoprepping smashing process, we can try and deep freeze them in liquid nitrogen, and they don't always freeze solid. What you end up with is kind of like a gum or a very rubbery material, and that doesn't smash very well. It just sort of ends up smearing in the bag. So this is something that we'll be doing a little bit of research into, is looking at the different kinds of collection methods. And as I said, this is something that I'm working on, because primarily flash freezing samples, it's very, very easy if you're in a lab. Very easy. You have all the materials, you have the equipment, you have big storage of liquid nitrogen or a big -70 freezer. But when you're halfway up a mountain and we have people working everywhere, it's much harder. The easiest thing to do is to take your sample and pop it in a tube and fill it with ethanol. Ethanol is a very good preservative for DNA, but it's not great when it comes to DNA extraction. So we're interested in different methods. So what we're testing in the lab are samples stored in ethanol, samples stored in something called DNA later, which again is just another preservative. We're looking at samples that aren't necessarily stored at -70 after collection. One of the interesting things about lockdown is lots of people couldn't send samples to us. We had individual people around the country who were having to store samples just in their freezer at home, in their kitchens, and that's only -20. So can we loosen our guidelines on how we're storing our samples? So that's a really interesting topic to look through, I think, and that's something that we'll be learning more and more about as Tree of Life gets going. You know, we've been going 18 months and most of that has been in COVID. We've already made some interesting progress, but there is a lot more left to come. And the same goes for the various extraction methods. We've found we've got great success with insects. Insects seems to be really, really easy. There's already very well established methods for tissue. There's been a lot of human research, a lot of research into mice, fish, and extracting DNA from those materials is very, very easy comparatively. But you start looking at plants, we have the problem getting through the cell wall, but then there are lots of polysaccharides. There are organisms with mucosoids. It's a very thick, gloopy material that's very difficult to wash off the DNA. You get lovely DNA at the end of it, but when you come to put it in the sequencer, it just blocks up the sequencer. You can't get any data from that. So that's another challenge that we're trying to overcome. And we can try that by using different kits. There are lots of kits that you can buy commercially, and there are lots of individual researchers in universities globally that are producing different methods that might be specific for their organism, but might also work for lots of others. So I think that's about as far as I get with my talk,
Perfect. Yeah, we've had a few sort of ideas coming from your poll around why different species need different extraction methods, and a couple that you spoke to. Someone put like the membranes are too thick, which is similar to what you were saying about plants and their cell walls. Someone asked whether in different species you may end up damaging the DNA.
Yeah, that's always a risk. One of the reasons why we're using things like the cryoprep, things like the grinding and lysis buffer, the more we can automate it, the more accurately we can control those processes. We found that if we do smash something too many times, we do end up fracturing the DNA, or we might not break the DNA initially, but we might just say, nick it, just break it just enough that it looks good. But then when it goes further down the line, it just falls apart, it degrades. We don't have anything useful at the end of it. So yeah, that is again, a very tricky part.
And then a couple of ideas that, yeah, you sort of mentioned in there, in that different samples might need to be preserved in different ways, which definitely depending on where you've got them from and what kit you have on hand. And because the quantity or quality of DNA might be different between different species. So lots of good ideas about, yeah, obviously different species are different for a reason. So how we deal with their DNA is also going to vary. We've had a couple of questions sort of specific what you were talking about before we move on to Petra. One question that I think is interesting, given what Petra's going to talk about, has asked how long would it take before a sample is considered like too old to be usable?
Ooh, that is a good question.
Does it matter? Like if it was flash freeze on collection, would it matter too much?
I guess the best answer I can give at the moment is we don't know. We have had samples that have been in our freezer for a year and they're still good. I expect that if they'd been in there maybe 10 years or 15 years, we might start to see a bit of degradation in there. But so far, I think everything that's come in has been pretty good so far. Not always perfect. There are lots of samples that come through and we have no idea why they haven't worked. But I'd say so far we haven't found a limit for storage in the freezer.
So I guess it's probably more to do with how well it's been stored rather than sort of actual age, right? Because I guess that a sample that's a few days old but hasn't been preserved is probably worse than a year-old sample been in the freezer. So it's probably a trade-off between preserving and age.
Yeah, exactly. And I think there's probably a bit of a link between the size of the sample as well. Something that's much larger is going to defrost or thaw much more slowly than something that's very small. And I talked a bit about the kind of the weighing and the dissecting of samples. all the time that those samples spend out the freezer, there's a risk that they get defrosted, there's a risk of damage to that DNA. So there's as much about the handling in the lab as there is about the storage.
Awesome. And then one last one for you, Michelle, before we hand over to Petra. Someone's asked, do individuals or companies voluntarily send you samples, or do you only take samples from sort of people within your team or people that you've sort of recruited?
So my understanding for this, I'm not part of this, I'm not part of the samples collection at all. But my understanding is we have a number of, I would say, sort of named partners. So we're in collaboration with Kew Gardens, Royal Botanical Gardens, the Marine Biological Association, but we have also received samples from scientists and companies elsewhere. So the Natural History Museum sends us a lot of samples as well. We've had samples from the University of Nottingham, from the University of Leicester. There was an individual collector in Northern Ireland that sent us some beautiful sea slugs. So we do have a variety. I think as well, especially through our Twitter, we've been trying to engage with the academic and research world and just sort of say, you know, suggest things for us. And I'd be willing to bet that if something was particularly tricky to get or was very, very specific, we would almost certainly get in touch and say, please send this to us. And I think also it's quite interesting as well, is who owns the specimen. Because at the end of the day, somebody collects that specimen, but initially it belongs to the person who owns, or the company that owns the land it was collected on. That is then transferred to the collector and then ultimately to us. And there is a lot of legal work behind there that sort of covers who owns that specimen and what happens to it. And I think ultimately we won't accept anything unless we have sole control over that specimen. Because at the end of the day, if somebody in 10 years' time says, oh, we need whatever's left of this hoverfly that was sent to you in 2019, if we destroyed all of that, it's much, much harder to go and retrieve that. Or even if there's just a tiny bit, digging that out of the archives is a lot of work. So we do tend to make sure that we have those samples at the end of the day.
Awesome, great, thanks so much for that. There's been a couple of questions that are quite sort of more general, and I think Petra might end up answering some of these in her talk, so I'm going to leave them for now. We can pick up any other questions at the end, but thanks very much for that, Michelle. I love the fact that freezing stuff and smashing stuff is an accurate part of science. Petra, we will hand over to you to hear about what you get up to.
All right. Go to the correct presentation. Okay. I will not be able to. I was keeping an eye on the chat and the questions, and I do like that some do connect to this talk. So Michelle very nicely explained what you do if you have what I would call the perfect samples. They're freshly caught, they're stored immediately in perfect conditions, and you get nice whole chromosomes out of them. So now I'm going to flip that completely and talk to you about, well, what if you can't do any of that? Because your samples are really old. No one thought about storing them for DNA. DNA wasn't even a thing when some of these samples were collected. So you have to make do with the samples you have. So some of you might have heard of the subgenre of genomics called ancient DNA or paleogenomics. It's when you dressed as if you were working on infectious diseases, but this whole attire, everything is not to protect you in any way, shape, or form. This is to protect the samples, because you're working on ancient bones or teeth or some other extremely precious historic sample that is fragile, is limited. You will get into a lot of trouble if you destroy it in any way, shape, or form. So just to reiterate, if you are working with present day organisms like humans, you can then look at specific organs, specific tissues, cell types. You can look at bacteria and viruses that live in and or around humans. If you are working on old samples, you don't really have that much left. You have some scattered DNA, you have some proteins, occasionally you even have lipids, but you just have to figure out what to do with what's there. On top of that, talking about degradation, the DNA through time, so you start with nice long chromosomes, through time, primarily due to water and oxygen and a whole bunch of chemical reactions as those two catalyze, your DNA will fragment. It's going to accumulate backbone nicks, and it's going to accumulate base modifications. So your DNA is going to look different than the DNA it started as. This is all issues that we can kind of deal with in post-processing once we get the sequences. But the biggest problem is, for example, this Neanderthal skull here has been sitting in dirt for hundreds of thousands of years. Your DNA sample is going to be full of contaminant DNA, primarily soil microbes, maybe some DNA from the people that excavated this bone. So it's not going to be the most ideal DNA extract. In most cases, microbes and humans have pretty different DNA, so you can clean out the microbial DNA. But what do you do if you have a Neanderthal and you have present day human contamination? That makes your life way more complicated, because Neanderthals and humans are sister groups. Their DNA is quite similar. And so going back to the question that was posed in the chat about a bit ago, how old is the oldest sequence DNA? And I put the organisms of each year bracket to kind of give you an idea.
So that poll should have popped up on your screen, but if not, pop an answer or any thoughts about why you picked what you picked in the chat box as well.
I can't vote, even though I wanted to put something.
So we've had no votes for mummies, we have a couple votes for Neanderthals, we have a lot of votes for mammoths, and quite a lot of votes for dinosaurs as well. So it's a pretty split between the last three, which is interesting.
Okay. So, oh wait, I have to remove the poll from my screen. So the oldest sequence DNA we have, which is reliably shown to be from the organism that the bone comes from, is a 1.2 million year old mammoth. The DNA came from a tooth that was found in permafrost. And permafrost is this layer of permanent ice, kind of comes in the name. So of permanent ice that you can find all across Russia and Canada, like at the very northernmost part of the country. It's basically a giant freezer. Your organism gets encased in ice, and well, due to climate change, more and more of the ice is melting, and so we're finding more and more organisms encased in there. The other years here are actually, so 4,000 is the oldest mummy we have sequenced. 430,000 is the oldest Neanderthal. So at this point, Neanderthals weren't even a thing. So this is like a proto-Neanderthal. We have sequences from that. Unfortunately, the dinosaur thing, kind of unlikely we're going to find any in permafrost, because permafrost wasn't a thing back then. There was many cycles of the earth completely melting and then refreezing and then completely melting in between. And the problem is DNA, like a lot of other molecules, has a half-life. So you start with long molecules of DNA, and then a half-life means that after this many years, you will have half of these molecules left. And then after this many years, another half, another half, another half. You probably heard the term in radioactive particles. So it's the same principle. And after a specific amount of time, you will have no original molecules left. And for DNA, it is theorized based on models, based on lab experiments, that DNA can survive up to 1 million years. And after that, you're going to have single nucleotides. Of course, this is DNA at less than favorable conditions. Like I said, this mammoth tooth was found frozen in a block of ice. Again, dinosaurs are very old. We're talking 65 million years. So, what kind of materials can we get DNA from? Bones and teeth are the most perfect DNA sources if you're looking at ancient DNA. So if you're looking at thousands and thousands of years, because they contain minerals and the DNA binds to those minerals, so it's a bit more protected. Like I said, you have permafrost, which is a big freezer, and it protects the DNA. In this case, the DNA that's bound to the bone minerals is protected also from water, oxygen, and further degradation. You can also get DNA from coprolites, which are fossilized poop. You can get DNA from hair. You can get DNA from other things found at archaeological sites, such as seeds, jewelry, various wood ornaments, and so on. And if you go a little bit more recent in time, you also have a lot of historic DNA collections, which are more or less all of our museum, natural history museum collections. So you have insects, you have various vertebrates, you have herbariums, and the organism that I work on is old mosquitoes. And when I say that, what people will very often picture is, of course, the amber encased mosquitoes from Jurassic Park. And so my question here is, do you think we can get DNA from amber encased mosquitoes?
I put that poll up as well, so it'd be interesting to see what people think to that. Interesting. It was very split at the beginning, and now yes is winning quite a lot. About 70% of people are voting for yes.
Okay, I feel like I made the questions really hard. So the answer to this is unfortunately no. The reason for that is amber is really, really old. So the youngest amber we have in the world is 16 million years old. All other amber is just going to be older than that. And as I said, half-life of DNA goes down to 1 million years. So after that, we won't really be able to find a sequence. It's just going to be single bases. And at this point, amber, everything in amber is pretty much down to carbon level. It's perfect for morphology, and it's perfect for preserving structures, but it's really bad at preserving DNA molecules. So this was my biggest disappointment, because I kind of wanted to do genetics because of Jurassic Park.
I presume if you had a mosquito and put it in amber and then took it out again after like a year or so, you might get something. But for then
it's not amber. Yeah, it's not amber. So amber is a tree sap, and it has various stages. I don't know the names of the different stages because that's more organic chemistry / geology. But there is things like proto-amber that is still kind of soft. And it's not yet the amber that we all know. Maybe unclear, but yes. So the actual samples I work on are pinned malaria transmitting mosquitoes. So you have tiny, tiny mosquitoes on a pin with these little labels that come with them, which tell you where was it collected, who collected it, and when. And then we can trace back, oh, this is a location where today we have a very diverse population of mosquitoes, or a very similar population of mosquitoes, or all of the mosquitoes in this population are insecticide resistant. What happened? So our aim is to sequence them and look for clues. Interestingly enough, for mosquito names, if you ever go and research what mosquito names mean, they're very often Greek and they never mean anything good. So malaria transmitting mosquitoes are from the genus Anopheles, which means useless. It's just going to be variation of that. It's useless, it's annoying, and everything in between for mosquito names. So this is a collaboration with the London Natural History Museum. The few of you that I've seen that are from the island might have visited it back in the day. And it's a collaboration with the senior curator of the mosquito collection, Dr. Erica McAllister. She's really funny on Twitter. If you don't mind occasionally seeing weird maggot photos, I would definitely suggest that you follow her, because you get some really, really interesting fly facts. So yeah, how does this, so I was in the beginning, you saw the picture of an ancient DNA lab where you work with fossils and you have to dress up and you have to be extremely careful. Since these samples are not bones that have been sitting in the ground, they're not full of microbial contamination, they're not full of present day mosquito contamination, you can be a bit more relaxed with them. You still need to be careful. So this is the old insects clean room that I set up at the Sanger. It's a small lab. You have everything you need in there. You have separate corners, which you can UV treat, and UV light basically breaks any kind of contaminant DNA. So you clean everything with UV, and then you can work on your samples. But the most important part here is that I put a sign on the door saying, this is a historic Diptera flies, historic Diptera lab. If you have done any present day fly work, please do not enter. So Michelle, you probably should never come to this lab after your long, nice chromosome DNA extractions. Because I'm trying to keep out any kind of present day fly contamination, especially since a lot of the work we do is also on present day mosquitoes, because we want to track how these populations are still changing and still evolving today. So there's a lot of present day mosquito DNA flying all over the place. I don't want any of that in this lab. And also the DNA extraction itself is very, the idea is similar, right? You have a lysis buffer, you put your sample in there, you then purify the DNA out of the lysis buffer, and you have a nice clean DNA extract that can go get sequenced. The actual procedure is fairly different. I am being extremely careful with these samples. They're really old, they're up to a century old, they're brittle, very dry. So my first step is I rehydrate them in just a styrofoam box with some wet paper towels. So they soften up a bit, they become less brittle. Then I gently dip them in lysis buffer. This is the picture on the right. The reason why one of the tubes is blue, that is not a typical color for mosquito tissue extraction, is because I'm also extracting the pin. Some of the pins used for pinning these old mosquitoes are really terrible. They just fall apart. The pin itself falls apart in the lysis buffer. But in any case, very gentle extraction. Then we take the mosquito out and we dry it and return it to the collection. Because again, these are limited samples. No one's going to go back to the 30s and the 40s to collect more samples. So we need to be very careful with them. And this is why we worked on this minimally morphologically destructive DNA extraction, where this is the same sample before extraction on the left and after extraction on the right. So you can see that the sample lost a few legs. It's a bit less colored. You can especially see that the eye pigments are gone. So the eyes are translucent now. But you can still clearly define morphological features on this mosquito. So all of the necessary bands are there, all of the necessary hairs are there, and you can still reliably say, yes, this is this and this species based only on the morphology. And so it might look slightly battered, but it's still good enough for entomologists to go back and look at. And now we're back to the DNA. We're back to some questions. So on this figure, you have present day mosquitoes, and this is the amount of DNA in nanograms that we got from present day mosquitoes. So it goes from like 30 nanograms up to 450 nanograms. How much do you think we get from historic samples? So going down to some of our oldest samples are from the 1920s.
So far, no one's gone for the biggest answer. We've got most people split between the lower two. So lots of votes for less than 50 or 50 to 150. Quite a few votes for 150 to 300. But the bottom two are wrestling out for the most votes.
That's pretty good. Most of the samples are under 50 nanograms. However, you can see quite a lot of dots above that. So we do have 50 to 150. Some of the younger, older samples, this is 1980s, actually do go up to 200, rarely. But yeah, so you do get less DNA from present day samples. But still, normally, if you, for example, tried to sequence these really old samples that are so low in DNA with the same methods you use for present day samples, they would fail. But because we know that the DNA is lower quantity, we can modify the procedure and still be able to get sequences from this. They're not going to be as great as present day samples, but they're going to be good enough that we can still look at them and compare them to present day samples. And the next question is about DNA length. So I said there's less total quantity of DNA, but what about the length of the DNA? So again, this is length in base pairs. Full chromosomes, in this case, are above 10,000 base pairs. They're typically like above 40,000 base pairs, which I kind of spoiled that part. But yes, how long is the DNA in these samples, do you think?
So the second answer seems to be winning so far. So most people are going for 200 to 1,000, but there's votes for all answers, but that one's definitely winning.
Okay. In this case, you're kind of all right, because I didn't specify how old the sample is. Now remember, the DNA degrades. It becomes shorter and shorter and lower in yield. So by that logic, the older the sample, the shorter the DNA. In this case, so this is the length of the DNA on the x-axis, and I put the important numbers, so 200, 1,000, and 10,000 in dashed vertical lines. So sample from the 30s, all of the DNA is below 200. A sample from the 60s, there's quite a lot of DNA below 200, but you still have long DNA up to 1,000. And then a sample from the 80s has DNA all over the place. It can even go up to 10,000. So in this case, depending on the age of your sample and depending how it was stored, you can get from really short to really long DNA. But again, we did modify the lab methods, the lab approaches to getting the sample sequenced. So it's fine with short, low yield DNA. And yeah, so we actually used all of this for sequencing about 200 historic malaria mosquitoes. This is a project that we dubbed Project Neandersquito, which confuses people. This is probably why I get the comments about Jurassic Park mosquitoes. In any case, this is a map of all of the samples that we have across sub-Saharan Africa In the bottom, you have a timeline. So the samples go from 1920s up to the 1980s. And they're from locations where we have present day mosquito sequence. So we can compare these really old ones to their great grandchildren living in the same locations today and see how things changed. Specifically, we are interested in insecticide resistance because that's fairly easy to look at, and that was one of the biggest pressures that humans put on these mosquitoes. But there's other things, and it might also be just human moved populations as well. And with that, I'd like to thank the rest of the Neandersquito team, and I would like to thank you for listening. Now I can actually look at the questions. And
thanks so much, Petra. And I love the difference between both of your sort of methods, where obviously, Michelle, you can get the best samples out there, but you have to deal with all different types of life, whereas Petra, your samples maybe aren't the best, but at least you get to concentrate on just the one mosquito there. So if anyone's got questions for Michelle or Petra, or just more broadly for the both of you, I'll make sure you're both on the screen, and we'll pick some out to ask. I know Michelle has been typing some answers in the box there as well, so I'm going to see if I can pick out a few. One that somebody asked that, Michelle, I've seen you type an answer to, but might be a nice one to hear a little bit from both of you, is what's the most common challenge you face in extracting DNA?
So I think the answer I put is really, we have kind of two main challenges that we look at, and that is getting enough DNA. The more, you can't have too much DNA. The more you have, the more you can sequence, the more data you can get. And the second challenge being getting really high quality DNA. Longer fragments of DNA are really, really good, because when you sequence the whole fragment. If you break that up, you then have to reassemble that programmatically afterwards. So the longer the fragment you start with, the easier the reassembly is later. So those, I would say, those are the two main challenges that we're facing.
And what about you, Petra? What's your biggest challenge?
for me, the biggest challenge is if it's the Natural History Museum in London, giving them back a sample that looks good enough that they can return it to the collection. So we do have other collaborators. We also collaborate with a lab in southern France that also provide us with a lot of the present day mosquitoes we work on. The challenge there is they like working on really weird cryptic species. This is something I didn't go into here, but mosquitoes like to hybridize. You think you have, this is a big problem in biology. What is a species? Never ask a biologist that. I've been working on that for 15 years. I still don't know. So mosquitoes like to hybridize, which means you think you have 3 species. In reality, you have 3 mosquitoes that like to mix and match, and then giant portions of one species chromosomes are in another species and things like that. So in that case, you get a mosquito that is supposedly one thing, turns out it's a completely different thing. And then you're, okay, did the person in the 50s just not check properly, or did things change so much in the last 50 years or so? Yeah, that would be the 2 main ones.
And I guess at least if that happens with you, Michelle, then hopefully you can just ask the person who collected it why they brought you a fish and not a hoverfly. Whereas, yeah, time's not on your side, Petra, in terms of asking why it was recorded as a certain species. Someone's asked a question specifically for you, Petra, asking wouldn't the rehydration step affect the water sensitive wings or body parts of those insects?
Yeah, so that's a good question. When I say rehydration, I mean specifically water vapor. So we don't dip them in water. This is why I put everything in this chamber. I put a lot of wet paper towels, and I put the little pin mosquitoes outside. So not outside of the box, but outside of the wet paper towels. Close it and leave it for 3 hours. And so the water vapor basically rehydrates gently all of the wet tissues. And you can really see some some of the really old samples, they look really brittle. Like I have to be very slow at handling them, and they still can lose a leg or two in the process. After that, I can just remove them from all of the labels and dip them gently in buffer, and they're fine.
So basically they get to go to the sauna before they get to go to the spa.
This is why it's called a spa. It's a full treatment.
Wonderful. Someone's asked sort of for both of you, but it might be something, Michelle, you might know more of, whether either of you have worked with DNA samples from marine animals, and whether there's any different challenges with marine samples.
Ooh. So I certainly haven't done old DNA samples. Sorry, I've just spotted the question. We have had some DNA samples. We've had starfish, lots of seaweeds. We've had a few mollusks, sort of shelled organisms, and what else have I seen recently? Oh, and I talked about sea slugs earlier. One of the challenges with those, especially for things like the mollusks and I say some of the mollusks and seaweeds, is we get those contaminants I was talking about earlier. So these polysaccharides, mucosoids that interfere with the processes later on. And yeah, we have just had a few that just didn't seem to work first time round. We've started separating our plants out into a separate plant protocol. They get treated slightly differently. And I think we've had some of our marine organisms have just worked. So we don't quite know what's going on. We do have somebody, Adele in my team is looking into these just to see what's different, what's going on, and can we tweak our processes just to make those work slightly better.
Awesome. And Petra, I don't know if you know anybody working with like ancient marine samples?
I actually extracted some DNA from historic fish.
Awesome.
So there is work. It comes with very different challenges. The problem with historic fish, for example, is the way they were typically stored, because again, back 100 years, people didn't know about DNA, they didn't care. They cared about the morphology. They would dip these in formalin. Formalin is very, very, very bad for your DNA. It's going to introduce mutations. It's going to introduce nicks. It's going to introduce, so you know how DNA has 2 strands? It's going to introduce binding in between those 2 strands. Your DNA is going to be basically bound to itself. You need to untangle that to be able to sequence it. So that's additional problems. That means it makes it more complicated to actually extract the DNA. You can't be very gentle with this. But again, a fish is a bit bigger. They're fine with you cutting like a tiny piece of gills or something and extracting that. But then also how you prepare the DNA for sequencing has to be different. Yeah.
Sounds like there's lots of challenges for all ancient DNA, just in different forms.
Again, if it's any kind of vertebrate, go for bones, because like I said, bones have minerals. The DNA can bind to those minerals and preserve better and longer. It's not marine animals, it's mummies, but for years, people have had a lot of problems getting good sequences from mummies because they were sequencing muscle, skin. And a few years back, one of the latest papers where they had a lot of mummy sequences, they were all done on bones because you could get good DNA from bones. So yeah. I don't know if that answers the question. I've never extracted a invertebrate, a historic invertebrate from the sea.
You're on mute.
Would help if I unmuted, wouldn't it? Someone's asked, do you ever extract mitochondrial DNA instead of nuclear DNA?
That one's fun. I think it's very difficult not to extract them simultaneously. So what we, certainly on my side at least, we extract everything. What goes in the tube is what it is extracted. So with these whole specimens, we're getting we don't get just a hoverfly, we don't get just a bit of frog. We get the organisms that they carry as well. So we get lots of bacteria, we get the mitochondria. We've got there's the Aquatic Symbiosis project that is a sort of, comes under the umbrella of Tree of Life. And they are specifically looking at organisms that are not just a single living entity, but things living together as a symbiont. And so from my perspective, I just extract what I'm given. I can't separate these out. What happens is they all get separated, it all gets mixed up and sequenced, and then it's the bioinformatician's job to pick that apart and reassemble those as not just one genome, but it could be several genomes. And yeah, and we already found multiple strains of bacteria that either we didn't know about or just weren't expecting. So it's a really interesting, I think it's a really interesting question.
Awesome. And Petra, I don't know if it comes up.
Yeah. So a lot of times when you work with really old samples, the mitochondrial DNA is pretty much the only one you can get, because there's, for example, in my mosquitoes, I get from 1 to 9x, which means I see on average a position in a genome 1 time or 9 times. So I have 9 sequences on that position, or I have only 1 sequence. For the nuclear DNA. For the mitochondrial DNA, same samples, it goes from 20 to 1,300. So I have 1,300, same sample that only gives me maybe 5 times a nuclear position gives me 1,000 times the mitochondrial position, because there's just so much mitochondrial DNA. It's much shorter, and there's just hundreds of copies per cell. You have a single copy of the nuclear, hundreds of copies of the mitochondrial. So in the end, you're just going to have a lot of the mitochondrial stuff.
So that was definitely a great question.
Yeah.
So we'll wrap up there, because that's just coming up to 5:30. But thank you so much, both of you, for joining us. I loved seeing the differences between your 2 works. And Petra will be joining us again next week to look more about her work in terms of the evolution of malaria, mosquitoes, and the medicines that we use to treat them. But thank you very much, everyone, for coming along. The link for signing up for next week's session will be on the screen in a second, and in the email that you'll get tomorrow as well. But thank you both for joining us, and thanks to everyone for tuning in.
Thank you for having us.
Thank you. Have a good evening, morning, afternoon.